This will be the first of our focused review or mind map sessions, beginning with Lab 1. So what we will do is we will go through all of the topics that are on the lab list and briefly highlight each one and review each one according to what we covered in both lecture or lab. The first topic that we'll examine for the Lab 1 lab list in this mind map session is how the different blood types will behave when mixed with anti-sera. So in the lab, when we're examining blood and trying to determine blood type, what we would do is place a drop of the blood to be tested on a card and then mix that blood with a liquid called an anti-sera. And the anti-sera, or the anti-serum, which is singular, contains antibodies against specific blood types. And if the specific blood type is present that anti-serum will detect, then what will happen is the antibodies in that anti-serum will cause that particular blood type to clump together. So much like we would see with a transfusion reaction. So to start off, we have anti-A anti-serum, which will cause blood with the A antigen to clump. So that is, if we test A-type blood with the anti-A anti-serum, we would see clumping. Same with anti-B, so the anti-B anti-serum will cause blood with the B antigen to clump. And we do not have anti-O anti-serum because individuals with O-type blood, or the O antigen, their blood will not clump in the presence of either anti-A or anti-B. What that means is that by process of elimination, we would see that O-type blood does not clump in the presence of any of the anti-A or anti-B anti-sera. So if we see that, we can, by process of elimination, determine that that blood has the O antigen. We also have an anti-serum for the Rh factor, because the Rh factor is simply another antigen on the surface of the red blood cell. We call that anti-Rh anti-serum. We also call it anti-D anti-serum. And if the Rh factor is present, that will cause the blood with the Rh antigen. If the Rh antigen is present on that blood, the anti-Rh anti-serum will cause that blood to clump. In individuals who are negative for the Rh factor, we will not see clumping when that blood is mixed with the anti-Rh anti-serum. So to visualize this a little bit, this is a very helpful figure in that it shows at the very top, the blood that's being tested is type AB, and we see that on the left side of the test card there is the anti-A anti-serum, which is typically a bluish color, and on the right side of the card is the anti-B anti-serum, which is typically a yellow in color, but I've also seen it having no color at all. It's a completely clear anti-serum. And what we see is that in the presence of the anti-A and the anti-B anti-serum, that both of those samples have clumped. And it's a little misleading in this diagram because the diagram is pointing at those two and it's saying that those are red blood cells. What they're actually indicating is that the red blood cells have formed clumps. And so what we're seeing in that image is not one single red blood cell. We're seeing a bunch of red blood cells, many red blood cells that have clumped together. So we see that type AB blood clumps in both the presence of anti-A and anti-B, which makes sense because type AB blood has both the A antigen and the B antigen. Moving down, the second from the top there, we see type A blood will only clump in the presence of the anti-A anti-serum, but it remains smooth and unclumped in the presence of anti-B anti-serum. Likewise with that, type B blood will remain smooth in the presence or unclumped in the presence of the anti-A anti-serum, but it will clump in the presence of the anti-B anti-serum. And on the very bottom there we see the type O, which has neither the A or the B antigen. So what that means is that the type O blood will not clump in the presence of anti-A, and it will not clump in the presence of anti-B. And we see the same thing for the Rh factor, where the Rh factor will clump in the presence of the anti-Rh factor if the Rh factor is present. And it will not clump in the anti-Rh anti-serum if the Rh factor is absent. So next we'll examine what the chemicals called heparin and histamine do. So heparin is an anticoagulant that prevents blood from clotting spontaneously during inflammation reactions, because what we end up seeing during inflammation reactions is that blood vessels dilate, and in doing so, blood flow slows down. And when we get to the module where we talk about blood pressure and the blood vessels, we see that dilation of blood vessels and that slowing of blood flow is what causes blood to start clumping and forming clots. So heparin helps to prevent clumps from forming during normal inflammation and inflammatory reactions. Histamine helps to dilate blood vessels. So histamine helps to expand those blood vessels during inflammation reactions, and that's actually what causes inflammation. And the dilation of blood vessels makes it a lot easier for leukocytes to exit the blood vessels to fight infections. So we saw that the leukocytes have the ability to leave the blood vessels to fight infections in the tissues. We called that ability diapedesis, and those leukocytes can perform diapedesis much easier if the blood vessels are dilated, because that also slows blood flow down, so that permits them to more easily leave the blood at the site of infections or where potential damage has occurred to the tissues and what could potentially lead to infections there or potential pathogens getting into the body from that site of damage. In the lab, we also saw that that the capillary tubes that we took samples of blood into to determine hematocrit, as well as look at the layers of blood, were heparinized. That is, they had a fine coating of heparin inside of them to prevent coagulation, because when blood is not flowing, it tends to spontaneously form clots, and so we want the blood to still be able to work with the blood in its unclotted form, and so the heparin helps to prevent clot formation. So here we see that when we remove samples of blood in the laboratory and place them in test tubes, we can spin those samples of blood after we put them in a heparinized tube. We can spin those samples of blood in a centrifuge, and what the centrifuge will do is it will pull the heavier elements, the formed elements like the erythrocytes, the leukocytes, and the platelets toward the bottom of the test tube, and it will leave the lighter liquid elements like the plasma at the top of the tube. And so we see that when we spin the blood sample at a high velocity in a centrifuge, we get these three layers. At the bottom, we get the erythrocytes, which are usually about 45% of the sample, and we call that the hematocrit because we define the hematocrit as the percentage of red blood cells in a sample of whole blood. And because it is the most dense of the blood components, it is pulled to the bottom when we centrifuge it. The middle coat, or the middle layer, is called the Buffy coat. It consists mostly of the leukocytes and the platelets, and it represents less than 1% of a sample of whole blood. And finally, towards the top, we have the lightest of the elements in the blood, which is the plasma and all of the things that are dissolved within the plasma, like the nutrients and the wastes. And that composes a little bit over half, about 55% of that whole blood sample. So how did we determine hematocrit in the lab? So what we ended up doing was we would collect a small sample of blood in a very thin tube that was called a capillary tube, and it was heparinized. So that is, it had a fine coating of heparin on its inner surface. So that would prevent the blood from clumping and coagulating when we were trying to determine hematocrit. So what we ended up doing is we'd fill that little capillary tube with a small sample of blood, and we would seal the capillary tube at both ends and then spin it in a centrifuge for about 10 minutes at about 4,000 to 5,000 RPMs to get to the heavier elements like the red blood cells to the erythrocytes to be pulled to one end, and the plasma and lighter or less dense elements to remain towards the top. And then what we would end up doing is we would measure the height of the packed red blood cells and compare that to the height of the entire sample. And that would give us a percentage, and that percentage represented our hematocrit. So let's look at some typical values and ranges for some of the attributes of blood. We'll start with hemoglobin concentrations. So hemoglobin is the protein in the erythrocyte that's carrying oxygen. So the measure of the amount of hemoglobin in the blood gives us kind of an indirect measurement of how well the blood is able to carry oxygen. So hemoglobin concentrations are typically measured in grams of hemoglobin per 100 milliliters of blood. For a healthy adult female, that's roughly 12 to 16 grams of hemoglobin per 100 milliliters of blood. In a healthy adult male, that's roughly 13 to 18 grams of hemoglobin per 100 milliliters of blood. Now, if we're talking whole blood volume, we measure that in liters. A typical healthy adult female has about 4 to 5 liters of blood in total. That isn't just including the hemoglobin and the red blood cells, that's also including the plasma. So it's including everything we saw in the previous slide a few slides ago. A healthy adult female has about 4 to 5 liters total blood volume. Healthy adult male has about 5 to 6 liters total blood volume. In both healthy adult males and females, we see that a typical number of erythrocytes is about 5 million erythrocytes per cubic millimeter or per microliter of blood. Those are the same units. So we have about 5 million in a very small volume of blood, which is important because the erythrocytes represent the blood's ability to carry oxygen, and so we want that to have to be the most numerous of the formed elements that we see. We also see, kind of based on the numbers of red blood cells, is the hematocrit ranges, and we give those in percentages, right? So a healthy adult female should have about 42% hematocrit. That is, they should have about 42% of their blood volume as red blood cells. And that number could be plus 5 or minus 5. So that could be a hematocrit as low as 37%, because that would be 42 minus 5, or as high as 47%, which would be 42 plus 5. So that gives us the average and range. Healthy adult males should have about 47% hematocrit. That is, their blood should be about 47% erythrocytes, plus or minus 5%. So that could be as low as 42% or as high as 52%. We'll examine next the general blood characteristics. Blood is a connective tissue, of course, composed of cells, which we collectively refer to as the formed elements. So those would be the erythrocytes, the leukocytes, and the platelets, which are not actually whole cells, but they are fragments of cells. And the extracellular matrix, which is referred to as plasma. So recall that connective tissues are composed of cells and extracellular matrix material. Plasma, which is the extracellular matrix of blood, contains mostly water. About 95% of plasma volume is water. And the dissolved substances that are in it mostly consist of proteins like albumin, the globulins, as well as fibrinogen, as well as nutrients. We elaborate on each of those substances and what their overall functions are in the lecture, so please refer to the appropriate lecture videos to review the functions of those. The blood itself, that is the erythrocytes, function to carry respiratory gases like oxygen, so the erythrocytes are carrying those using the protein called hemoglobin, and the blood is also carrying carbon dioxide, but not a lot of red blood cells carry carbon dioxide. Most of the carbon dioxide is actually converted into another chemical called bicarbonate that is dissolved within the plasma. Looking at the functions of blood, we see that blood performs important transport functions like transporting respiratory gases to and from the lungs, transporting nutrients, typically from the digestive system to all the cells and tissues, because the cells and tissues are going to be using the nutrients that are obtained from the digestive system. And the blood is also transporting metabolic waste typically that are produced at the produced at the tissues and taken to the kidneys for removal. The blood also transports hormones as well as the endocrine system, and the endocrine glands will secrete their chemical messenger hormones into the bloodstream, and the blood will carry those hormones throughout the body. Blood also has a couple of regulation functions as well. It regulates body temperature. It regulates its pH, that is an aspect of its chemistry, using mostly substances like bicarbonate. So bicarbonate is what's responsible for making the blood slightly alkaline and keeping the pH at a slightly alkaline level. And the blood also regulates its fluid volume using proteins like albumin. So again, I mentioned that there's a more elaborate discussion of what the blood proteins do in the Module 1 lecture material. Blood also has some protective functions in that it protects us from infection by microorganisms like bacteria, viruses, from tumors that arise from our own cells and tissues, or from parasites. Blood also protects us from hemorrhaging or blood loss by coagulating and clotting. And the entire process of coagulating and clotting is outlined in the Module 1 lecture material as well. We'll also look at some disorders of erythrocytes. Most of the erythrocyte disorders that we're going to examine fall under the category of anemia or anemias. And anemias are disorders of erythrocytes in which erythrocyte numbers or hemoglobin is low. So we have a couple of different examples of some fairly common anemias. We have things like hemorrhagic anemia, which is a low erythrocyte number due to lacerations, ulcers, or massive blood loss. So hemorrhaging is simply a general term for blood loss. Iron deficiency anemia is also fairly common, and it results in the low production of hemoglobin because iron is required to produce hemoglobin because it's iron that helps to carry oxygen. And we also have pernicious anemia, which is a vitamin deficiency anemia. We get low production of erythrocytes due to low availability of vitamin B12. Vitamin B12 is a really important vitamin in helping with cellular mitosis, in creating new cells. And one of the cells that's really, really dependent on it are the stem cells, the precursor cells, that will eventually become the mature erythrocytes. So having adequate vitamin B12 is important for continued production of new red blood cells. We also have renal anemia, which is low production of erythrocytes due to low secretion of a hormone called erythropoietin. Erythropoietin is a hormone that is produced by the kidney, and its job is to essentially stimulate the red bone marrow to produce new erythrocytes. So when production of that hormone is low, that means that the stimulation of the red bone marrow to make new erythrocytes is also going to be low. We also see, finally, hemolytic anemia, which hemolytic refers to the premature destruction of red blood cells. This could more commonly be caused by transfusion reactions because mismatched donor and recipient blood types will cause the recipient's immune system to create transfusion reactions to destroy the mismatched blood. Or malaria parasites can also cause hemolytic anemia because the malaria parasite, or part of the malaria parasite's life cycle, involves replicating itself in red blood cells. And when those red blood cells get so filled with the parasites, they literally rupture and are broken and destroyed. Both of those situations represent situations in which red blood cells are destroyed. We also have some focus on the leukocytes as well, what the leukocytes do and what their names are and the types of infections that they are designed to fight. So first we'll take a look at the granulocyte leukocytes. So these are the neutrophils, the eosinophils, and the basophils. These are the leukocytes that have small granules in their cytoplasm that are visible when these cells are stained to be viewed under the microscope. The neutrophils are a very common leukocyte that help to fight acute bacterial infections, so things like appendicitis or bacterial meningitis, things like that, strep throat, so bacterial infections that are not recurring. Eosinophils are next, and eosinophils specifically kill parasitic worms that enter through the skin, the lungs, or the digestive system. But also we see that eosinophils are responsible for producing inflammation reactions in the lungs due to inhaled irritants. So those are one of the types of leukocytes that can stimulate asthma attacks because of their inflammation reactions. And we also have, finally, basophils, and basophils function in producing heparin and histamine during inflammatory responses, which are two chemicals that we discussed earlier that help to really control various parts of the inflammation response that is carried out by the immune system. And finally, we will end our discussion of the Module 1, or Lab 1 lab list, with the agranulocyte leukocytes, which are leukocytes that do not have dark staining granules in their cytoplasm. There's two types. We have the monocytes and the lymphocytes. The monocytes are similar to the neutrophils, but they fight chronic bacterial infections, whereas we saw the neutrophils are fighting acute bacterial infections. So chronic bacterial infections are things like TB, tuberculosis, or chronic viral infections like hepatitis, or certain types of leukemias, which are blood cancers. And finally, we see the lymphocytes, in which there are three types. We have the NK cells, which are also called the killer T cells. These are specific types of lymphocytes that will kill cells in which it does not recognize the glycocalyx of that cell. So often what we get when cancers or tumors develop is that the glycocalyx of our own cells that have become cancerous will change their glycocalyx, and the NK cells, or the killer T cells, will specifically target and destroy those. They will also target virus-infected cells as well, because virus-infected cells in our body will often change their glycocalyx as kind of a warning marker to the immune system that something's not right in that particular cell. We also have B cells, which you can think B as an antibody. So B cells produce antibodies to fight against very specific types of viruses or bacteria, even years after first exposure to those viruses or bacteria. So these are the cells that are producing the antibodies that help us in our kind of long-term resistance to certain types of infections. And then finally we have the T cells, which are the cells that actually go after the antibody tagged cells, because the B cells are producing antibodies that are going to tag specific pathogens or viruses in our body, and the T cells are those cells that are actually going after those antibody tagged bacteria or viruses to destroy them. So that concludes our Module 1, Lab 1 focused review mind map session. Please feel free to reach out to me by email if you have any follow-up questions, and I'd be happy to help. Thanks.